The overall goal of this procedure is to analyze whole zebrafish embryos between the 4-cell stage and 32 hours post-fertilization for the presence and location of apoptotic cells. This is accomplished by first dechlorinating embryos at the desired time point. Next, the embryos are fixed and permeabilized. Then, the embryos are incubated with rabbit anti-activated caspase-3 antibody, followed by a fluorescently tagged anti-rabbit secondary antibody to fluorescently label apoptotic cells. Finally, the embryos are analyzed by fluorescence microscopy. Ultimately, results can be obtained that show differences in number and location of apoptotic cells between control and treated or mutant embryos through immunofluorescence.
The main advantage of this technique over existing methods, like analysis of apoptotic cells by acridine orange staining or the tunnel assay, is that the caspase-3 assay is a highly reproducible and relatively inexpensive method for experiments in which photographic documentation and/or quantitation of apoptotic cells is required across multiple samples. Demonstrating the procedure will be Shelley Sorrells, a graduate student from my laboratory, and Christian Taruno, a technician from my laboratory.
Under a chemical fume hood, prepare 50 milliliters of 4% paraformaldehyde, or PFA, by adding PFA powder to 1x PBS. Shield from light and stir at 60 degrees Celsius for 1 hour to dissolve. Store at 4 degrees Celsius for 1 month or at -20 degrees Celsius for 2 years. Prepare 100 milliliters of 4% methyl cellulose by heating 30 milliliters of water to 80 degrees Celsius. Then add 4 grams of methyl cellulose and stir to dissolve at room temperature 4 hours to overnight. Bring the volume up to 100 milliliters and mix at 4 degrees Celsius for 1 hour. Store at room temperature. Collect embryos immediately after fertilization and let them develop until the desired time point between the 4-cell stage and 32 hours post-fertilization, or HPF. For embryos between tailbud stage and 32 HPF, remove the chorions by incubating embryos in 100 microliters of 10 milligrams per milliliter pronase in 10 milliliters of 1x egg water for 30 minutes at room temperature. Pour off the pronase solution, slowly add 1x egg water and repeat the rinse 2 more times. Then transfer up to 40 embryos to a 1.5 milliliter tube and use 1 milliliter of 1x PBST to rinse 2 times. After removing all but about 100 microliters of the PBST, add 1 milliliter of 4% PFA. Lay the tubes on their side in an appropriate size container for ease of transport and place on a gentle rocker at 4 degrees Celsius overnight or up to a week. After fixing the embryos, remove most of the PFA solution and carefully, in a dropwise fashion, add 1 milliliter of ice-cold 100% methanol. Lay the tubes on their sides and place at -20 degrees Celsius for 2 hours. Alternatively, embryos can be left at -20 degrees Celsius for up to 24 hours. After the incubation, allow the embryos to settle to the bottom of the tube before removing most of the methanol, and carefully adding 1 milliliter of 1x PBS with 0.3% Triton X and 1% DMSO. Immediately, remove the PDT and replace with fresh PDT, then lay the tubes on their sides and rock for 30 minutes at room temperature. After carefully removing most of the PDT, add 500 microliters of blocking buffer and, ensuring that the embryos stay submerged in solution, gently rock for 1 hour at room temperature. Next, add 1 microliter of rabbit anti-activated caspase-3 antibody and additional non-rabbit antibodies if desired. Gently rock the embryos for 2 hours at room temperature or overnight at 4 degrees Celsius. After removing the antibody solution, add 1 milliliter of PDT to quickly rinse the embryos. Then replace with fresh PDT and gently rock for 30 minutes at room temperature before repeating the wash. Remove the PDT wash and repeat the blocking step. Then, add the appropriate fluorophore-conjugated secondary antibodies. Shield from light and gently rock for 1 hour at room temperature. Under a fluorescent microscope with the appropriate filter, analyze the embryos in 1x PDT. If positive controls have bright fluorescence with minimal background, then transfer them to a 3.5 cm petri dish with a thin layer of 4% methyl cellulose, or a depression slide with a large drop of methyl cellulose. Using an angled probe, orient an embryo of interest for imaging, taking images that represent fluorescence intensity and patterns observed in the majority of the embryos from that sample. This figure demonstrates that injection of embryos with high concentrations of mRNA encoding potent pro-apoptotic BH3-only proteins, such as BIM or NOXA, quickly gives rise to active caspase-3 by the 4-cell stage of development. Embryonic death subsequently occurs within 1 hour. At later stages of development, fluorescence intensity is more robust. For example, shown here are bright active caspase-3 positive cells in sphere stage embryos that have been injected with moderately toxic levels of mRNA encoding the pro-apoptotic BH3-only gene PUMA. Messenger RNA encoding the anti-apoptotic BCL2 gene was co-injected to prove that injection of PUMA mRNA induces apoptosis through the intrinsic mitochondria-mediated pathway. IR-induced apoptosis in zebrafish requires p53-mediated transcription of BH3-only genes such as PUMA. The delay in IR-induced apoptosis shown here is consistent with the requirement for p53-mediated transcription. The caspase-3 protocol should give rise to a robust signal-to-noise ratio. This figure compares the optimal caspase-3 assay result to two common suboptimal results that arise when critical steps are compromised, as outlined in the discussion section of the text protocol. First is a weak signal-to-noise ratio due to background fluorescence in animal tissue, and second is yolk fluorescence that distracts from the signal of interest. Finally, as shown here, co-labeling experiments using transgenic GFP-expressing lines approximate tissues undergoing apoptosis. The transgenic oleg-2 EGFP VU12 line labels motor neurons and oligodendrocytes, and the transgenic ELAV-L3 EGFP line labels all differentiating neurons. The results suggest that both neurons and oligodendrocytes are sensitive to IR-induced apoptosis.
Following this procedure, other methods like co-staining with tissue-specific primary antibodies and embryo sectioning can be performed in order to pinpoint the exact identity of cells undergoing apoptosis.